Good morning, everyone. My name is Angelo Morales. I know we've all introduced ourselves to each other 6 weeks ago when this class started, but nice to be having somewhat of a more personable conversation with y'all, seeing faces, hearing voices as we do these presentations. So I look forward to hearing all of your presentations in the next couple of days as I listen to your thoughts on these different topics and look forward to being able to hopefully teach a couple of things in this presentation and get some constructive feedback as well. So I will be speaking about receptor tyrosine kinases and its interaction and contribution to cancer, and that is going to be the goal of this discussion. Although this is a graduate level course and everyone in this class is well educated and very intelligent, I do recognize that biochemistry is a complex field. And I know that I am personally still developing my understanding of many of these concepts, and they are complicated. So this presentation, my goal is to have the material explained in a detailed yet approachable manner. I want to use clear, straightforward terminology whenever possible. I want it to be able to consistently and accurately break down the steps of these biochemical processes and the concepts that we can all understand without insulting anyone's intelligence and without sacrificing any sort of scientific accuracy in terminology. So that is my goal. I hope by using more accessible language and providing context for important terms and mechanisms throughout this presentation, I hope we make all of ourselves, I hope we all become a little bit more comfortable with this topic. Please do excuse any mispronunciations I have. I did do a very thorough research on this topic and looked up a lot of the pronunciations for some of these pathways, but I am not an expert. And if any of you have constructive feedback on how to present some of these terms better in the future, please let me know in the comments y'all leave for me next week. Um, During this presentation, we're going to look at how RTKs normally work. We're going to look at what happens when the signaling goes wrong, how certain cancer treatments are designed to target these abnormal signals, and what would prevent these targeted treatments from being effective. So that's the goal for this presentation, how normal growth signaling becomes a non-oncogenic driver and a therapeutic target with RTKs. So let's get started, guys. Okay, so here's the roadmap for today's presentation and the learning objectives we're going to have during this conversation. So first, we want to look at the basic structure of what receptor tyrosine kinases are, what RTKs are, and what happens when a signaling molecule attaches to that receptor. Then we're going to move on to two important pathways inside the cell. They're known as the RAS-MAPK pathway, which helps tell cells when to grow and divide, and then the PI3K AKT pathway, which helps cells survive, use energy, and then grow. Next, we'll discuss how cancer cells can take advantage of these normal signaling systems and how RTKs can thus become oncogenic. We'll walk through all those steps. Then we'll look at, finally, the HER2 and EGFR as real-world examples and discuss how these medications and how certain medications can target these receptors to treat cancer specifically. We'll see how these cancer cells can then sometimes adapt and become resistant to these changes. So this is going to be the goal as we walk through, kind of keep these bullet points in your mind as we're checking them off. If we understand each of these learning objectives, we're going to be successful in understanding the material presented in this class and what RTKs do in the human body. All right, so RTKs, receptor tyrosine kinase. What are they? RTKs are proteins that sit across the cell membrane on the outside of a cell and help communicate with their surrounding environment. According to one of my references, you can find at the end, if you have questions on it, do in Lovely, there are 58 known RTKs in humans. Although these receptors respond to different signals, they all have a relatively similar basic structure. The outside portion of the receptor acts almost like antennae. It detects signals such as growth factor and then calls these signals in, so to speak. These receptors then pass through the cell membrane, connecting into the cell, and they transfer information in that method. On the inside of the cell is the tyrosine kinase region. When the receptor is activated, it uses ATP to attach phosphate groups, and these are in specific areas of the receptor. And we can think of these phosphate groups as docking stations where other signaling proteins can come in and attach. This starts a chain reaction that carries the message deeper into the cell and can eventually tell the cell to grow, to divide, or to continue surviving. Normally, this process is very carefully controlled. RTKs themselves are not harmful, and that's one of the key notes of this presentation. The RTK itself does not cause any oncogenic factors or effects. The problem occurs when these RTK receptors become overactive or basically stay turned on. The switch is on when the switch should be off. And when that happens, the cell continues to receive signals to grow and to divide. It continues, continues, continues. And as we all know, this continuing signaling is what contributes to cancerous cell growth. So this is, in a very, very succinct little slide, what an RTK is and how it functions inside the cell. All right, so normal RTK activation pathway. It is relatively complex, but if you take it step by step, it's understandable. So the process begins when a signaling molecule on the outside of the cell, and once again, please excuse me for mispronunciations, but due to all my research, I believe the correct pronunciation is ligand. So this ligand on the outside of the cell attaches to the receptor, and it's almost like a key fitting into a lock. So these receptors are coming in and attaching to that ligand. Once the ligand binds, two receptors come together or change their position, which activates the portions of the receptors located inside of the cell. The receptors can then add phosphate groups to each other. And these phosphate groups basically, once again, are docking sites. So they're where other proteins inside the cell can attach. The important thing to understand is that the RTK itself is not doing all of this. Instead, it's kind of like a switchboard. Once it's turned on, it can activate different communication pathways within the cell, which causes these other cells to be able to come in and dock. These pathways can tell the cell to continue to grow, to divide, to use energy, to survive, or even to move in certain situations. So the problem that arises is if the RTK becomes overactive, it can send too many of these signals at one time. This is one of the ways cancer cells can take advantage of a normal cellular process and turn it into uncontrolled growth. If that RTK is sending too many signals and creating too much growth, we will have an overproliferation of abnormal cellular growth. And here we come to some of the signaling pathways that are activated by RTKs. And once again, this slide looks very complicated and it looks confusing. And it is a very in-depth biochemical concept. But when you take it step by step, it is relatively easy to understand. So on the left is the RAS-MAPK pathway. Think of this as a chain reaction where one protein in turn leads to the next. As the signal moves down, it is eventually causes the proliferation in the case of RAS-MAPK, or survival and growth with PI3K. So as I was saying, the signal moves from RAS to RAF in the RAS-MAPK pathway, and then on to MAK and finally ERK. ERK is the final step in which the nucleus can be entered, and genes can actually be influenced by these signals that are being passed down this pathway, basically almost a game of telephone. And this tells the cell when to grow and to divide. On the right side, we see the PI3K, AKT pathway. This pathway is especially important for helping with the growth, survival, and metabolic use of energy within the cell. AKT can also activate another important protein called mTOR, which helps regulate metabolism and protein production. So these two pathways, don't work completely independently. They communicate with each other as well as with a much larger signaling network within the cell that is all producing these end results that we see. For us and for the purposes of this class, the important takeaway is that a cancer cell needs more than just the ability to divide quickly. It can divide all at once, but if it, at the end of the day, doesn't have an avoidance to cell death, the ability to obtain nutrients, the ability to survive when stress is placed upon it, it's still going to die off. When RTKs stay turned on for too long, they're continuously activating pathways and giving cancer cells advantages at the same time by providing all these um, opportunities for cell growth, along with the avoidance of all those other factors I just mentioned. All right, so RTK signaling, as we understand, can lead to that development of or overdevelopment of cells, which can lead to cancer. So RTK signaling needs to be turned off to avoid that. For normal signaling to remain useful, it must both be activated and terminated at appropriate times. You just can't have it on all the time. You can't leave the light switch on all the time. That will have negative outcomes. Cells have several ways to turn RTK signals off. If this was an hour-long presentation, we could get a lot more into depth in regards to how these systems work. However, just due to time constraints, I'm going to be brief with these. So protein tyrosine phosphates can remove phosphate groups. Activated receptors can be internalized from the membrane and either recycled or degraded. Cells can also use negative feedback proteins to reduce signaling intensity. Downstream tumor suppression is also important. For example, the PI3K pathway is opposed by PTEN, which helps prevent excessive AKT signaling. The key idea of all this is that normal signaling is temporary and context dependent. A growth signal should be active when the cell is trying to receive the correct stimuli, and then it needs to diminish or turn off when that stimuli is gone. Problem is, cancer frequently disrupts this balance. If a receptor remains active, or if the downstream pathway becomes independent of the receptor, the cell can behave as if it is continuously receiving instructions to grow and to survive. So to sum it up, we basically need the RTK signaling to be on, so we can imagine the light switch on when we want this process to be working effectively, but it needs to turn off. It needs to diminish when the cell no longer needs it. Otherwise, we're going to continue the signaling pathway, which is going to lead to unwanted outcomes. So now that we understand these previous processes as outlined so far, we can now look at how RTKs become specifically oncogenic or cancerous. On this slide, we see four major ways that RTKs can become so overactive they lead to this end result of a disease state. Although it looks like a complicated lineage or different mechanisms, they are relatively easy to understand. Once again, I think as I continue to study these subjects and I see it broken down, they are easy to understand when you get to the basic explanation of each of these. So the first method or mechanism by which we can have this persistent growth and survival signaling from RTKs is mutation. A mutation can cause the receptor to become stuck on the on position. Once again, going back to the analogy we used of a light switch, it's on. There's no disruption of that signal. So even when there is no normal signal telling it to activate, we're continually telling these cells and these signaling pathways to continue that growth. Secondly, we see gene amplification, also known as overexpression. And this simply means that the cancer cell makes far too many copies of a receptor. So as we'll see in a minute in the next slide, HER2 positive breast cancer is a good example of this. Cancer cells can have unusually high amounts of the HER2 receptor, which is promoting that gene overexpression. Thirdly, we see chromosomal rearrangements. In this case, pieces of DNA can become rearranged and they can create abnormal proteins that are continually sending growth signals. ALK and RET are examples that can be affected in this way when the DNA itself is sending rearranged signals. And then finally, there is the autocrine activation, also known as the autocrine loop. So this is basically when a cancer cell produces its own growth signal and then responds to that same signal. So it's creating a self-feeding loop where it encourages the cancer to keep growing and it's feeding on itself. At the end of the day, though, what you need to understand is that even though these four mechanisms are different, the results are similar. The normal on and off switch, which is effective in reducing that signaling, becomes disrupted. And this allows the cancer cells to continuously receive signals to grow and to survive and spread. And we'll look at that a little bit more in this next slide. And here we arrive at the core of the presentation. How abnormal RTK signaling produces cancerous growth. So as we see here, we're explaining how this overactive RTK signaling has such major effect on this proliferation of cells. As we've already discussed, when that RTK is staying turned on, it sends out several signals at the same time that can help the cancer cells survive and grow. The RAS-MAPK pathway encourages the cell to grow and divide, while the PI3K AKT pathway helps the cell avoid normal programmed cell death. The AKT and mTOR pathways can also help cancerous cells use nutrients and make the proteins they need for rapid growth. RTK signaling also affects how cells move and attach to surrounding tissues. So this can help cancer cells invade nearby tissues, not just the parent, so to say, organ or tissue. In some cases, this can rapidly spread to multiple areas of the body. Cancer usually involves several different genetic changes. So RTK signaling is rarely the only problem seen there. However, some cancers become extremely dependent on that one abnormal receptor or signal pathway for their survival and proliferation. We sometimes call that oncogene addiction. This is important for treatment because if we can identify and block the specific signal that a cancer depends upon, we may be able to significantly slow or stop its growth. And that's the basic idea behind targeted cancer therapy, which we will discuss in these following slides. And here we see how this affects us in real world scenarios. So this is HER2, HER2 or HER2 is a positive breast cancer. It's a good real world example of what happens when an RTK becomes overactive. HER2 is a receptor found on the surface of the cell. In HER2 positive breast cancer, the cancer cells have extra copies of the ERBB2 gene, which causes them to produce far too many of these HER2 receptors. With so many HER2 receptors on the cell surface, growth signals can be sent out much more frequently than is normally found. This activates pathways that we've already discussed, including the RAS-MAPK and the PI3K AKT, which encourage cancer cells to grow, divide, and survive. This discovery has had a major impact on how breast cancer is treated because doctors and researchers can test a patient's tumor to see whether it has too much HER2. And if it does, they can use medications specifically designed to target the HER2 rather than treating every breast cancer exactly the same way. And as we've seen after decades of research, cancers are rarely exactly the same, and you need specific targeted medications and treatments to be able to have positive outcomes. And this is a great example of precision medicine. identifying what is specifically driving a patient's cancer and choosing treatment based on that abnormality. It also shows why understanding these signaling pathways is clinically important. What happens at the cellular level can directly influence how we are going to diagnose, classify, and treat cancers. And we'll look at that targeting here. And here we arrive at targeted therapy. Forgive my pronunciation if I'm saying this wrong. If any of you on the clinical side have a better pronunciation, please let me know. But trastuzumab, also known as Herceptin, is a medication specifically designed to target the HER2 receptor. It's a type of drug called a monoclonal antibody, meaning it is designed to recognize and attach to a specific target. So it knows what it's looking for and it will attach to it. When trastuzumab attaches to HER2 on the cancer cell, it can interfere with the signals telling that cell to grow and to divide. It can also help decrease the amount of HER2 available on the cell surface. Another important effect is that trastuzumab can also help the immune system recognize that cancer cell. So not only is it finding the targeted cell, it's helping the body's own immune system recognize that cancerous cell. Once the antibody attaches to HER2, immune cells can recognize that antibody and attack the cancerous cell. This process is called, very long term, antibody-dependent cellular cytotoxicity, or ADCC. If any of you guys are jiu-jitsu fans, you also know what the other ADCC is. The bigger picture is that targeted therapies like trastuzumab try to make advantage of something that makes the cancer cell different from normal cells. If a tumor depends heavily on HER2 signaling, blocking HER2 can have a major effect of that cancer on that cancer. However, targeted therapy isn't always permanent. Cancer cells can adapt and develop other ways to keep their growth pathways active. And this is one reason a treatment may work very well initially, but eventually become less effective as cancer develops resistance. And here we have another example of an RTK that can contribute to cancer. So this is EGFR. EGFR belongs to the same family of receptors as the HER2 is, and is especially important in certain types of non-small cell lung cancers. In some lung cancers, mutations cause EGFR to become overactive. This means the receptors can continuously send signals telling the cancer cell to grow and survive, just as we previously discussed with HER2. Some tumors can become very dependent on this abnormal EGFR signaling, which gives us an opportunity to specifically target it with medications. HER2 and EGFR show us two different ways we can target these receptors. So trastuzumab is an antibody, as we've discussed, that attaches to the outside of the cell. Many EGFR medications, on the other hand, are small molecules that can enter into the cell and block the receptor's activity from the inside. You can think of these medications as the same type of faulty switch as the other locations. It's just turning it off from the inside. It's working on the inside. Instead of a breaker on the outside of the house, we have a switch on the inside. The important takeaway from this, the reason I present this, is that not every cancer has the same molecular abnormalities. By testing a patient's tumor for specific mutations or for abnormal proteins, clinicians can choose treatments that are more likely to work against that particular cancer. This is another example of how precision medicine is changing cancer treatment and research. So I already explained this, but this is just a synopsis. The two main ways of targeting RTKs. You have the monoclonal antibody method, which is the external outside part of the cell targeted. And then you have small molecule TKIs. And that is when these much smaller drugs are entering into the cell and blocking that signaling process from the inside. So both approaches have the same overall goal. They're shutting down the signaling pathway. But unfortunately, cancer cells will change over time. They're going to adapt. A treatment that initially may work very well may eventually become less and less effective as the cancer develops new ways to keep those growth signals active. And this brings us to one of the biggest challenges with targeted cancer therapy, which is drug resistance. And this is why targeted therapy can fail. Targeted cancer treatments typically work very well, but cancer cells aren't static. There's change, and there's an adaptation over time. Cancer cells can use different signaling pathways to get around basically any blocked receptor. If one pathway is shut down, the cancer cell will find another way to send the same signals via a different route. Resistance can also develop further down the signaling pathway. For example, proteins such as RAS or PI3K may become permanently active. If that happens, blocking the receptor at the cell surface may no longer matter because the growth signal is already being produced inside of the cell. And then finally, tumors are made up of many different cancer cells. Some resistant cells may already be present before treatment even begins. Therapy kills the sensitive cells, but resistant cells survive and multiply. This is similar to natural selection happening inside the tumor. It is also why cancer treatment may involve repeated genetic testing, combinations of different drugs, or newer medications designed to target the specific changes that allow the cancer to become resistant. So, to wrap all this up, to summarize everything we've discussed, RTKs are proteins that help cells receive and respond to signals from outside the cell. When a signaling molecule binds to the receptor, the RTK becomes activated and adds phosphate groups to specific sites. These sites then activate the signaling pathways such as RAS, MAPK, PI3K, AKT, which help control cell growth, survival, metabolism, and gene activity. Cancer, however, can take advantage of this normal system. Mutations, extra copies of genes, gene rearrangements, or abnormal signaling can cause RTKs to stay active, even when they should be turned off. Examples, as we've seen, include the HER2-positive breast cancer and certain lung cancers with EGFR mutations. In these cancers, identifying the abnormal receptor can help doctors choose a treatment that specifically targets it. This leads to the main idea behind precision cancer therapy: determine what molecular change is driving the cancer, and then choose a treatment designed to block that specific problem. However, cancer cells can continue to change, which means that they will eventually become resistant to that treatment. Overall, understanding RTK signaling shows us how basic cell biology connects directly to patient care. By understanding what is going wrong with these signaling pathways, we can better diagnose types of cancer and select more effective treatment, helping with more positive outcomes and lives drastically affected in a very, very amazing way. Here are the main references I utilized for this presentation: our textbook, along with some great peer-reviewed scientific articles. And I hope that the references help you if you have any further questions. And if not, please reach out in the comments this coming week, and I can discuss any other questions left after this presentation. Um, Thank you so much for your time and attention and listening to this presentation, and I wish you all a very good week.